Welcome everybody. I'm Alan Davis, for those who don't know me. I'm one of the geriatricians here and clinical director of Health Older People. It's been fortuitous today that we've had the opportunity to have some outside speakers to come to talk to us. I just first want to acknowledge the Brain Research New Zealand for sending us these people, and Alzheimer's New Zealand, who are represented down the back by Kevin Salmon and Rod for actually having arranged to get these people here to us today. So it's the pleasure I can introduce Lakshini Mendis and Helen Murray, who are researchers from the Brain Research Centre for Brain Research in Auckland, which is part of Brain Research New Zealand. Particularly to talk to us, they do a whole lot of things there, but particularly to talk to us about Alzheimer's disease, because it's Alzheimer's month. Brain Research New Zealand's taking the opportunity to get their researchers out into the countryside too. So meet with real people, and we can meet with real researchers and talk particularly about Alzheimer's disease. I've told Lakshini and Helen that we're a very diverse audience, so there's hopefully going to be some diverse information. Lakshini and Helen said just interrupt if you want to ask questions, and we will, I'm sure, have a good time with it. So I'll hand over to you guys. Thank you very much.
Thank you.
Yeah, we're just going to do a bit of a tag team sort of presentation, so Lakshini's going to start off.
Thanks. So thank you for having us here today. Thank you to Alzheimer's Northland, who together with BRNZ has sort of organised this whole day for us, which has been fantastic. So a little bit about Brain Research New Zealand, or BRNZ. It's quite a new initiative. It's a Centre for Research Excellence that's co-hosted by the University of Auckland and Otago as well. Let me just start my time again. And so we have about 67 different research groups spread out through the country, part of both universities, and also the University of Canterbury and AUT, so Auckland University of Technology. And we've got groups kind of looking at the brain from all different angles, very interested in sort of normal, healthy aging, but also neurological disease. So we're New Zealanders working for New Zealanders, and we've got this amazing opportunity to be able to, work to help New Zealanders right here. So Helen and I are actually based up in the med school in Auckland. It's there, and we're up on the 5th floor up there. Penthouse suite. That's a research team. So we're part of a group that's led by Associate Professor Morris Curtis. And the sort of primary goals of our research group is to understand the process of new brain cells sort of production in the adult brain, and sort of the neuroplasticity angle, and also to understand how it changes in neurological diseases such as Alzheimer's or Parkinson's disease. And this is a shot of the lab. So the Centre for Brain Research is where we're at. And the Centre for Brain Research sort of brings together groups across the University of Auckland, diverse groups working from sort of looking at psychology, audiology, movement neuroscience, lots of different angles again. So the main goals of the Centre for Brain Research, or the CBR, are to unlock the secrets of the brain, develop new therapies, and we're definitely based on sort of these 3 pillars. So we've got neuroscientists who work together with the clinicians, but also we've got good ties with the community. So it's sort of, all these 3 groups kind of working together to understand what's going on, I guess. So we definitely like to engage with communities. That's why we're out here talking to you today. And not only to sort of share with you the research that we're doing, but also to understand sort of what questions, I guess, you want us to focus on, because I think that's really important as well. And so at the CBR, we've got various different modalities that we use to unlock the secrets of the brain. So obviously, MRI is a big one because you can understand real-time changes that occur. And recently, we've just started let's see what it's called, the dementia sort of research clinic And the idea behind that is to get a cohort of people sort of at a mild cognitive impairment kind of stage, so that you can then follow these people on, look at biomarkers, look at the changes that you can have, so that we can sort of better, I think, identify the process of Alzheimer's disease, but also look at sort of novel biomarkers and things like that to help us understand the disease at an earlier stage. So we've got other facilities as well. We look at growing new brain cells in a dish, looking at what happens. This is a really neat video showing you real live brain cells doing their thing. We really like that.
Are they thinking?
Well, we don't. Yeah,
Are they thinking?
these ones are actually Parkinson's. Well, don't quite know. But the really neat thing about being able to do this is that we can then, we have links with sort of medicinal chemists who supply us with sort of novel compounds and things like that. So we can add it to a dish, see what they're doing to these cells. You know, are they making them move a little bit faster, make those connections a little bit stronger? What's happening there? But yeah, so usually you'll see cells like this, but you know, what makes a neuron, and cells like this, but you know, what makes a neuron, and that's definitely where some electrophysiology comes into it. So you're looking at it, you attach an electrode into these neurons, look at their sort of electrical pulses, and that's how you know that it's sort of a functional neuron. So we have groups doing sort of changing skin cells into you know, these neuron-like cells. And so this is a final stage to kind of make sure that it is what it is. A lot of what Helen and I do are imaging. So we look at the postmortem human brain, and we look at normal brains and sick brain cells, and look at sort of what the difference is between the two. So we're both incredibly lucky to be able to have this awesome resource, the Neurological Foundation Human Brain Bank that we have up there. And we have gifts sort of from all over New Zealand. We're supported by a number of community groups. And it's all under, you know, informed consent from the donors, but also their families. And yeah, for me, I think there's a sacredness to being able to work with postmortem tissue, and it's a very precious resource we have. So we have about 242 normal brains up there, which is really important because that's sort of our reference point for what, you know, normal healthy aging looks like. And then we have tissue from sort of a range of different diseases. And it's a little bit different about the way we do things in the brain bank, because we then organize it according to sort of different functional blocks. So both of us look at the hippocampus because we're you know, interested in Alzheimer's. But what this means is it kind of extends the use of this resource. And we've got tissue from sort of one of the first brains that were donated to us. So that really highlights sort of, you know, the scope and for this resource to kind of be applied for different projects and things like that as well. So that's a little bit of a background about who we are and what we do, but we're here to talk about Alzheimer's disease. So I'll hand over to Helen now, and then kind of jump in at the end and talk a little bit about what I do.
Can you hear me? Cool. So I'm just going to go over a little bit of background information about Alzheimer's disease, just so everyone's on the same page for when we sort of talk about our research. So Alzheimer's disease is one of the leading causes, or is the leading cause of dementia. It accounts for 60 to 80% of cases, dementia cases in total. And worldwide, the number of Alzheimer's cases is projected to double every 20 years. So that is a huge number of people that are affected by the disease, and the associated cost of care with that sort of increase is very large as well. So we believe, obviously, Alzheimer's is, and the prevention and treatment of Alzheimer's, research into that is incredibly important. Just some sort of facts, sort of looking at it. Age is a key risk factor. With any normal aging, there's accumulation of pathological proteins and aggregates. So age is sort of the key risk factor for Alzheimer's. The age of onset is around 65 years. This does vary a fair bit, but that's sort of the general age of onset. It does tend to be more prevalent in women. And symptoms, everyone is sort of familiar with the memory loss associated with Alzheimer's disease, but there's also a number of other cognitive and personality and mood changes that are associated, problems with language, judgment, abstract thinking, and many others, and some of which we're going to talk about in terms of our research. Things like the loss of smell, which is not something that people typically associate with Alzheimer's disease, but it's actually one of those things that does occur. So to put things in context, this is a normal, healthy human brain. It's got lots of tightly packed convolutions. There's not a lot of space. If we cut it down this sort of plane here and had a look at it face on, this is what we would see. There are, obviously, hopefully everyone sort of knows a little bit about brain anatomy, but there's this grey matter on the outside and there's white matter on the inside. So the cells, the neurons themselves, live in the grey matter, and then the white matter is essentially the highways that their tails will then go down to project to other areas. So everything is quite tightly packed. The only real space are these fluid-filled ventricles. And then we compare to severe Alzheimer's disease. You can see it's incredibly shrunk. There's obviously a lot of space going on, and this is due to the loss of huge numbers of neurons all over the brain. Most people only sort of think about the hippocampus, which is this area down the bottom, in terms of loss of memory, but there's actually cell loss all over the brain, and it produces thinning of the grey matter and also the white matter as the cells die and their tails, the highways down to the connections also die. So there's a lot of space going on here, and obviously this is a very, very sick brain. If we look under a microscope, this is sort of the classical pathology that we see with Alzheimer's disease. We have these aggregates that form inside of neurons called tangles, and they're made up of a protein called tau. And compared to sort of, this all happens in normal aging, but in Alzheimer's disease it's just so much more pronounced. We also have these amyloid plaques, and this is sort of one of the things that gets put out a lot in Alzheimer's media and everything, that these plaques are really, the bad thing that's happening. And these are sort of clumps of this protein called amyloid that form outside of the cells. So any sort of clumping or aggregation of proteins is not good for the brain. And to have this much of it, we think, is one of the key things that is leading to these cells dying and producing that huge atrophy that you see. So now I want to just talk about what I've been doing in my PhD. So as Lakshini said, our lab group is really interested in looking at plasticity in the human brain. And so my project has primarily focused on the Alzheimer's disease brain and using that human postmortem tissue that we had from the brain bank. So first of all, what is plasticity? This is sort of a term that sometimes is not that well understood. It's the ability of the human brain to adapt and change. It's what underlies our ability to learn new skills, to make new memories, and to repair or compensate for a loss of function after an injury. So it's a very important function that we have and that we want to see if it's changed in Alzheimer's. So there are ways that the brain can go about adapting and changing. It can make new neurons. This is something that for a long time people thought the brain couldn't do, but we now know that it definitely can make new neurons in adult life. Neurons can also change their structure, the way they're branching and the way that they're connected to other neurons and how they communicate with those other neurons. So these are all mechanisms that the brain has at its disposal to be plastic. And what I've done in my PhD is sort of have a look at each of these three things and to see whether it's still functional, that process is still functional in Alzheimer's disease. Because if it is functional, then it gives us the ability to perhaps boost it to try and repair some of the damage that's going on. And if the process is severely impaired, then maybe it's not the best therapeutic avenue to take. So I think it's quite relevant to potential treatments in the future. So to start off with, I'm just going to sort of introduce you to this process of making new neurons and what we found with that. So neurogenesis is this process of making new neurons in the human brain. And in the adult brain, it only occurs in two specific regions. They both sit very deep within the brain. So if we sort of cut where this blue line is, this is what we would see if we looked at a face on. this red line is what we call the subventricular zone. It's because it sits underneath this big fluid-filled ventricle. And it's one of the regions of the human brain that can create new neurons. And it overlies the basal ganglia, which is one of the key structures involved in movement. But the new neurons don't actually end up in here. They actually migrate all the way down to the base of the structure and then forward in the brain out to the olfactory bulb, where they're involved in the detection of odours. So this is something that's particularly interesting for us when we're studying Alzheimer's disease, because the loss of smell, or anosmia as it's called, is one of the first symptoms to occur in a number of neurological diseases, including Parkinson's and Alzheimer's. It's one of the symptoms that people don't typically associate, but it happens to be one of these pathways where new neurons are produced, so there could be some relevance there. The second area that produces new neurons is extremely relevant to Alzheimer's disease. It's the hippocampus. So this area down the bottom here, it's a little bit further back in the brain, is the key structure involved in the formation of new memories. It's also the area that is most severely affected by Alzheimer's disease, and we know that memory is a big problem. So if we look at the hippocampus a bit closer, there's this dark band of cells called the granule cell layer. This is an important part of that learning and memory circuitry. And then this black line here, just under it, indicates the subgranular zone, so beneath the granule cell layer. And this is where new neurons are produced in the hippocampus. And they migrate up into this blue layer and become part of the learning and memory circuitry. So again, the two regions where new stem cells are produced are actually highly relevant areas for Alzheimer's disease pathology. So what we did, we wanted to ask the question of whether the Alzheimer's disease brain can still make new neurons. And so we had tissue from 12 control and 12 Alzheimer's disease brains from the Neurological Foundation Human Brain Bank. These cases are carefully matched for age, gender, and a number of other factors that we get from the patients and their families. We then cut very thin sections and labelled them for a marker of proliferating cells, so we could see which cells were proliferating. And this is what we found. So the first thing was that there are far less cells in the hippocampus, new neurons produced in the hippocampus, than in the subventricular zone, which is sort of evident by the scale here. There's, on average, in a normal brain, about 1 new neuron every 5 millimetres of that subgranular zone. Whereas up here in the subventricular zone, there's about 20 cells every millimetre. So there's a big difference there in the amount of cells that are being produced. And it suggests that this is probably not a big source of new neurons in the hippocampus. So that was an interesting finding in itself. The second thing was that there is a very large variation between individuals. The amount of proliferating cells in any given person is quite different, which I guess is expected because we all lead very different lives. But the most promising thing that we found was that the Alzheimer's brain retains the ability to make new cells. There was actually no significant change in the hippocampus anyway between the number of new neurons that were being produced. So that is actually quite a positive thing, suggesting that the Alzheimer's brain, despite all that atrophy, is still producing new neurons. In fact, it was actually increased in the subventricular zone. So this is quite positive. So we wanted to look a little bit further, because to make a new neuron is just the first point. These cells then need to grow and migrate to where they need to be. So we know that they're being produced, but are they becoming healthy, normal neurons that are doing their function? So the second part of my project was looking at how these neurons change their structure and their connections to each other. And to do that, I looked at this protein with a very long name called PSA-NCAM. It stands for polysialic acid neural cell adhesion molecule. There will be a test at the end. And it's composed of these two parts. So there's the NCAM part and there's the PSA part. I'm going to stick with the abbreviation. So the NCAM is actually a protein that's expressed by all cells in the human brain, and they bind on to NCAMs from other cells, and in that way they keep the cells locked together in a nice tight spatial arrangement, which is very important in such a complex structure like the brain. But if you add this PSA onto the end of an NCAM, it can no longer make that connection. So it's essentially insulating the cell from its environment. It's what we like to think of it as like the detergent that you would put on yourself before you went down a slippery slide, because it reduces the friction involved in movement. And what it means is new neurons can express this polysilated form of NCAM and grow and change. So this is a very pretty image that I took very early in my PhD, highlighting this is what a normal, healthy, mature granular cell looks like in that blue layer that I showed you previously. You can see it's quite complex. It has a cell body and it has a lot of these branches at the top. It also has a tail, but I'll cut it off so you can't see it. And a new neuron looks something like this. So it's sort of round and spherical, slightly elliptical shaped. And this neuron needs to change from this sort of shape into this kind of shape. So by expressing PSA-NCAM, a cell can grow and remodel these long, complex branches. And if you look really closely, you can see there's these little spines along all of the branches, and these are actually the physical connections between cells. And so PSA-NCAM also allows the cell to remodel those connections between other cells. And that's really important when a cell is trying to integrate into a new sort of function or a new circuitry. The other thing, of course, is that it allows a cell to migrate. By coating themselves in this sort of slippery protein, it can then migrate to where it needs to be, which, as you saw for the subventricular zone, was actually quite a long way down out into the olfactory bulb. So again, we had a look at PSA-NCAM in our 12 cases that we had originally looked at proliferating cells. And what we saw was, again, very promising. So again, there's a large variation between individuals, which I think we sort of expected by this point. But the Alzheimer's brain actually had plenty of PSA-NCAM cells in those regions that make new brain cells. So actually it was no different to the normal brains that we saw. So this is suggesting that even though there's all of this incredibly large amount of cells dying, there's still a lot of plasticity happening in the Alzheimer's brain, which is very good in terms of looking at a potential way to boost the system to increase some sort of therapeutic output maybe. But during this study we actually found some other really interesting things. So this protein, PSA-NCAM, has only really been described in terms of these newborn neurons and how they grow and adapt. But this is an image of the hippocampus of my PSA-NCAM staining. And what we actually saw was quite different. So this layer here is the blue layer I showed you before. To make it easier, I've put a red line in. So this is the subgranular zone where the new neurons are made. But all these dots here are actually cells or neurons that have PSA-NCAM. So what we actually saw was that there are plenty of neurons all over the hippocampus expressing this marker of plasticity, which suggests that it's not just the new neurons that are incredibly plastic, it's all of, well, not all, but a lot of the neurons in the structure. And this is quite novel and interesting in terms of human research, so we thought we'd go a little bit further. And we took regions from all over the human brain. We took 10 regions in total, and they all have very different functions, all the way from sort of the superior frontal cortex, which is involved in personality and mood, down to the visual cortex, which is obviously vision. This is an area that's involved in memory. Down here is the hippocampus, so just sort of from the inside here, involved in learning and memory. The cerebellum and the basal ganglia, which are hugely involved in movement. So these regions all have very different functions. And we looked for this PSA-NCAM, this plasticity marker, and sure enough, we found it everywhere. So in these cortical regions, this green one and the red one up here, it was arranged into these layers, which corresponded quite well with where the cells themselves live. And it was mostly those branches that were expressing the protein. So that suggested that the branches in those regions are very plastic. In other regions, we saw whole cells. And they're quite beautiful. I'm a little bit biased, but this is my favourite of all time. And so the cerebellum has these very complex Purkinje cells which are involved in coordination of movement. And we saw, this was the one cell I saw in the entire cerebellum section I looked at. So this was a very specific type of plasticity that's being, that's undergoing by just certain types of cells in certain regions. In other regions, the cells look very different. And this is an area involved in movement, and it was particularly interesting because no one else had ever talked about PSA-NCAM in this region anywhere, in rodents, rabbits, anything. So to find these cells in the human brain was quite exciting. We also had a look at some numbers. We also actually looked at Parkinson's disease in this study, but I'll just talk about the Alzheimer's disease component. And again, it was very promising. So in the regions that we quantified, for most of them, the number of PSA-NCAM cells was about the same in Alzheimer's and control. So all over the brain, there's still this amazing plastic ability that's being retained by the cells that are still alive. We did see a significant decrease in this region called the entorhinal cortex. So this is the bit at the bottom of the hippocampus, but it's heavily involved in that learning memory circuitry. So the decrease that we saw there was quite interesting because this is also a region where those plaques and tangles that I showed at the start, where they first start appearing and where they're most severe at post-mortem level. And this is just a sort of a wide magnification view of that. Those layers that I showed you before, they're pretty much all gone in this region of the Alzheimer's brain. So this is a region heavily affected by pathology that has a huge decrease in its plasticity marker. We compared it to the amount of those tangles this correlates very well with disease severity. So more severe Alzheimer's have more of these tangles. And we saw this inverse correlation. So cases with more tau had less PSA-NCAM. And that was pretty consistent. So that's sort of just a very quick overview of some of the findings that we had, and just a very quick summary slide. We saw that in Alzheimer's, the brain still has this ability to make new cells, and this could be an interesting avenue, therapeutic avenue in the future. We saw that a lot of neurons in the brain are geared up for plasticity, and that this still happens in the Alzheimer's brain, which is also very exciting. And that in this one region of the Alzheimer's brain that's so heavily affected by pathology, there's a decrease in that plasticity. So whether that is a cause and effect, we don't know, but it's certainly interesting. So just to finish up on what I have showed you, I just want to comment on sort of what this means for you or for your patients when you're sort of talking about these sorts of things. My big picture point here is that the brain has this amazing ability to adapt, but it needs to be stimulated. To retain this capacity and to hopefully prevent something like Alzheimer's disease happening, we need to stimulate it. So how can we stimulate plasticity? These are just some of the things that there's been some good evidence to show which increase that. So physical exercise, doesn't have to be strenuous, just something. Social interaction, meeting new people, new experiences, and cognitive exercises like things like memory games or crosswords, whatever sort of tickles your fancy, but these are all things which have been shown to increase the amount of plasticity when you brain cell production. So I'd like to recommend that that's something that you pass on. Yeah, so that's sort of where I'll stop, and just to acknowledge a couple of people. These are a lot of people that worked on this project with me, and some funders. The Hope Foundation have been excellent in funding this work, and of course the Brain Bank for the amazing contribution which we never would have found this without. So I'll pass on to Lakshini.
Okay, so I'm just going to change track a little bit and maybe talk about something that's people aren't aware of, because it's quite new research as well, but it's out there. Published a paper quite recently, so you're free to go look up my name and read about it. But let's try to break this down. So I'm looking at the hippocampus in Alzheimer's disease as well, but I'm looking at lipid changes, which is a little bit different. So you have your brain cell, beautiful brain cell here, and I'm very focused on just that little part, or the lipids that make that part, your membrane. So this is biology 101, way back in undergrad. So your neurons and everything in your neuron is all compartmentalized and by a lipid bilayer, lipid membrane. And if you think about the fact that you have 1 billion neurons, I think is about the estimate. Lots of neurons. Each neuron has about 1 million of these little lipids. That's a lot of lipids. And so it's pretty accurate to say, oh, we're all fatheads because it's about 60% or something that we have lipids up there. So the lipid is, it has a polar head group and this long hydrophobic fatty acid tail, which is why it makes up this lipid bilayer. So you have the polar head groups that are outward facing, long fatty acid chains that are inward facing. And this is obviously a static image, but the lipids itself are very fluid. And that's sort of how it controls the structure of the neurons. And obviously we need it when we're talking about migration and things like that. It plays an important role. And they also make these sort of lipid raft areas, and that's to do, so the more cholesterol you have in there, the more rigid it gets. And we now know that it's sort of these areas where you have a lot of these integral membrane proteins, the ion channels, the receptors you get in there. So these areas are very involved in sort of, you know, a whole host of different functions. So play an important role. And everyone's sort of aware of the pathological hallmarks of Alzheimer's disease, so the amyloid beta plaques and the tau tangles. But in his seminal paper, Alzheimer actually describes sort of this accumulation of lipids that they found in these, you know, aging brain cells as well, that he thought might have an effect in sort of disrupting the structure of it and causing sort of cell death. So that's sort of where we come in. And because there are, you know, millions of different lipids, and you have, and each lipid can have a different polar head group, varying lengths of tail. So you're talking about sort of a lipidome that's more diverse than our proteome or genome. And so until quite recently, there wasn't a very good way of sort of analyzing or looking at particular lipids. So we'd be able to sort of stain for a lipid group, a certain lipid group, but it's only quite recently that we can look at sort of individual kind of lipid species, if you like. So looking at the human hippocampus, in the past, a lot of the lipid work that's been done, they'll look at sort of the hippocampus as a whole, because the technique kind of uses kind of mashed up little bits of tissue that you'd run through a machine. And so no one really knew a lot about how these lipids were distributed, which we think is really important, because you have all of these different areas even within the hippocampus as well. So Helen's touched on really nicely the granule cell layer, the subgranular zone, and there are also sort of, you know, the white matter tracks you can get, and these sort of subfields that each play a different role in that sort of memory circuit. So what we wanted to do was to image the distribution of lipids in the human hippocampus, and then try and detect sort of specific lipid, sort of see if there were any specific differences in these various subfields. So, in typical science form, we use a lot of jargon, really long words. And so this is a technique I'm using. It's called matrix-assisted laser desorption ionization imaging mass spectrometry. I'm just going to call Mal the IMS from now on. And so what I do is sort of use a bit of post-mortem human tissue, very thin, 12 microns, put it on a plate, put some matrix on it. So the matrix, not quite the movie back in the day, but this is sort of a low molecular kind of powder that we sort of coat the sample with, put it in our machine, and hit it with a laser. So this is basically what happens in the machine. As your matrix and your sample will get ionized, it flies through under vacuum, where it's separated according to its mass. So if it's a smaller molecule, it will fly faster. We detect it earlier on, and if it's heavier, you can detect it a little bit later on. So lipids are really tiny. They're about 300 to about 1,000 Daltons, which is, I don't know, tiny. I don't know how to compare it. So this is sort of the general idea, and then what we do with the imaging mass spec side of it is we scan our tissue. So I don't know if you can see, but there are these tiny little dots on the tissue, and that's where the laser has struck the tissue and ablated the sample. And so what you can then generate is sort of one of these spectra in at each of these points. then you do a whole lot of pre-processing, which basically for me as well is mathematical magic. Put it through the algorithms, run that, get this. But what you can, the really neat thing is you can then pick any of those peaks and look at sort of a distribution heat map of these. So these are just a few images that we can look at along there, and that's the hippocampus. And so you can then figure out what your hotspots are. Where are these, different lipids concentrated? And this is a really neat technique that other groups in the world are using to sort of look at things like tumor tissue, look at sort of tumor boundaries in the tissue, fingerprints to look at sort of, if this person's taking drugs or not, things like drug distribution in animal models, that kind of thing. So it's quite a versatile technique, and it's quite new in Auckland, so very lucky to be able to work with it and sort of try to develop it a little bit more. Right, so once you have this kind of information, what you can do is you can stain your tissue histologically and define kind of the regions and be like, tell me what my profile is for this region. Or you can look at sort of how these different spectra are grouped together. So taking the second approach, one of the big things we found was obviously the difference between grey and white matter. And this is just a couple of examples that came up. So this lipid is abundantly sort of expressed in the grey matter, and so that means it's probably involved in making the cell bodies. Whereas this one is coming up in the white matter, and so it's more involved with the myelin, the sort of projections, the communication side of things. And this is normal hippocampus, but it was really good to see that the distribution pattern was conserved in Alzheimer's disease. But you can probably already say, so with this, the more of this lipid that you have, the sort of darker, or the heat, it's a heat map, so the more red-orange kind of color indicates a higher abundance of this, whereas a blue will just be low abundance. So you can already see that there are some differences that you can identify just by eye of how these are differently expressed. And so one lipid group that we saw were these ones called the phosphatidyl ethanolamines. And you can see that it's especially in the CA1 region, which plays a role in sort of the memory circuit. That's one that changes. Oh. And these ones are sort of linked to, a lot of these ones were linked to polyunsaturated fatty acids. So those are your like omega-3s that a lot of people are talking about. So obviously you're losing these ones in the Alzheimer's disease brain. But there were others that were increased in Alzheimer's disease, such as your ceramides, which are kind of known to play a role in apoptosis, so it wasn't entirely surprising that this was increased in the brain, because apoptosis is, your cells are dying, it's a mechanism of cell death. And there were other ones, like the phosphatidyl serine species, that were also increased, especially around this sort of dentate gyrus region area. And again, that's sort of an indicator of sort of cell death and something's going on. The thing is, this is sort of a snapshot of end-stage disease. So we know that these things are changed at the end stage of disease. What we now need to go and do is to figure out if it's a consequence of disease or a driver of it. Because there's evidence for it being the latter as well. So this is what we know so far. We know that there are different lipid profiles. The profile itself is conserved in Alzheimer's disease, but it's expressed at sort of different levels and in very specific regions as well. So we still need to figure out if this is a consequence or a driver of pathogenesis. And to do that, it's probably, what we're going to have to do is look at other areas of the brain that are not quite affected at the end stage. So areas such as the visual cortex, or, I don't know, I can't think of any other thing. Yeah, everything's affected. Or just going back to sort of the cells we had in a dish and trying to add these different lipids, see how it's different. But what this work kind of shows is it gives us an idea of sort of that cellular environment and how it's changing in Alzheimer's disease. So it enables us to sort of better manipulate kind of what's happening as well. So when we create these models for Alzheimer's disease, we can create better models incorporating sort of the actual other changes that are happening. And then a big thing I always get is sort of external factors such as diet, because omega-3 is a sort of big highlight at the moment. So understanding how, well, if we're having, if we increase our omega-3 uptake, does that actually then have downstream effects? How does that change in the brain? And there are some really interesting work sort of happening with magnetic spectroscopy as well, where people look at sort of, it's sort of, I guess, like an MRI technique, where you're looking at these very specific sort of lipid groups. So that'll be a key to kind of follow through an early sort of Alzheimer's disease cohort, see what's changing there. Yeah, so I'd just like to acknowledge all of the people who are involved, funding bodies, people who helped me with the imaging analysis, lots of data to analyze, that was always and of course the Neurological Foundation Human Brain Bank. I think I'll leave it there. We'll open it up for any questions that you might have.
Any questions that people want to ask?
Helen, I'm a nurse. This might be a dumb question, but can you create that PSA-NCAM?
I think we can stimulate it. We've got a lot of work going on looking at how it's regulated, how it's produced, and how it gets removed. That's a big thing that's happening in our lab at the moment. So right now, we don't have a great way of increasing it. The best thing we've actually found is insulin. Insulin actually seems to keep it on the cell surface. But we have to be a bit careful, because we need to make sure that there's a good balance, because you can't have, if all of the little neurons kept their PSA-NCAM, they wouldn't then be able to properly make the connections that they need. They have to get rid of it at some point in order to talk to each other. So I think it's a balance between making more of it in this area that's obviously affected, but also keeping the dynamics right, keeping the removal right as well. We still need to answer the question of, what is it doing? Why are certain cells expressing it and not others? So there's definitely a lot more things to answer, but yeah, it's something that hopefully we can look at in the future.
Now you've found that in basically cadaveric samples, are you going back to look at potential animal models? And I'm actually thinking in terms of embryonic, and going through and taking samples with progression to see what changes?
It's funny you mention that, because I'm actually in contact with a guy from Washington DC who's looking at an Alzheimer's disease mouse model, which he has found starts in the olfactory bulb, which is where we think a lot of this pathology actually begins. It's an area that's connected to the outside world by a very thin layer, and we think that there might be some driver there where you get sort of a lot of these sort of environmental hits over time, which sort of leads to this accumulation of these bad proteins. And we know that's an area where PSA-NCAM is really important. And so I'm hopefully going to go work for him in the years coming. But yeah, we're starting a collaboration looking at exactly that, young embryonic Alzheimer's and how that might change as it goes. Because of course we can't do that with humans. It's something that we don't really have a lot of young human tissue donated. It's something, to be honest, we really need, because it's everything we do is end stage, aging, and to know what is aging and what is disease can sometimes be a tricky point. So you do actually need a young sort of comparison, which we don't really have, and it's a very difficult thing to ask for, but it's something we really do need.
And on that same vein, I'm thinking about your lipid work, you'd also need to have a look back at what's happening embryologically.
Yeah.
Because all you're seeing at the moment is end result.
Yeah.
That could be anything.
Yeah. Yeah.
There's a huge thing in Alzheimer's research at the moment that if you could prevent the onset of Alzheimer's disease by 5 years, you would lower the incidence by half. which is huge. And basically it's because if you can keep the brain healthy for longer, people will live a healthier life. They have a better sort of end stage, healthy neurological outcome, and essentially die of other things. So, at least what we're talking about is quality of life, really, right? You can prevent this for longer.
As a nephrologist, I'm asking these questions, I'd rather not actually.
But definitely, we do need to go back. So the thing with our group, I think as well, we don't have a good sort of working model like that in our lab. So those collaborations are really important. And because the lipid work I'm doing is sort of quite new in the field as well. So there are some people who've looked at sort of more animal models of the disease and that kind of thing. But it would be good to sort of be able to, go earlier on, see what's happening.
Because I'm also thinking in terms of the dietary side of things.
Yeah.
Whether you're changing it early makes a difference, if at all. Because I'm equally thinking by the time you've got the symptoms, it's end game, forget it. You're not going to actually change it.
Possibly, we don't.
So epidemiologically, have you been able to link with the lipid research as well?
Well,
because we know
I'm just thinking
lipids, epidemiological lipids are related to, lipid management is related to Alzheimer's.
Exactly, and there was sort of that recent study, so looking at omega-3s and stuff as well. So we found, there were studies that were really positive and sort of, animal models of the disease and things like that. And then there was a study looking at sort of like an Alzheimer's disease cohort and seeing if, omega-3s that would have an effect. And they didn't really find an effect, so I think what they're doing now is sort of going, well, if we can catch it early enough, so, to see if that will make a difference. And I think that's how a lot of the other work that I was talking about with the dementia sort of clinic that we have will come into play as well. So you're looking at biomarkers so that you can identify these things early enough. We're looking at, even things like your gene status for ApoE and things like that, which is not something we've sort of accounted for in the work that I've done. But that might play a role as well. Yeah.
David.
Thanks for a fascinating talk. The increasing numbers that are predicted, is that just a function of an aging population with more older people? Is it a diagnostic bias, or do we think there's an exposure that's driving a true increase in incidence once you stratify for age?
Possibly all three. But definitely aging population. I think that's the key driver behind that number. Diagnostically, it's actually very difficult to accurately diagnose Alzheimer's. You can't actually do it until you look at the pathology in the actual brain, because you need to see these plaques and tangles. So I'm not entirely sure if diagnosis is one of the big things, but it could very well lead to that number. Epidemiologically, I think there are things that are being identified as potential contributors to that build-up but I don't think there's anything that's really conclusively been shown as a big environmental factor that is leading to it. Yeah, aging population is the big one.
About a week or two ago, there was all that news thing about the research that was being done overseas with regard to the plaques and tangles, and are you associating with that particular educational institute at this point? And what's your name?
No.
Honestly, there's a huge amount of research that goes into plaques and tangles.
Because they were looking at the drug that actually prevent the plaques and tangles.
Oh, right, yeah.
So it was the drug that was depleting tau protein.
Yeah. So I was wondering how much effect that would actually have, because if you can deplete the tau protein, then you can actually see the cause, maybe see the cause and effect.
Yeah.
There's a lot of therapies going into doing that sort of thing. A lot of immunotherapy as well has been looked at. So far, nothing's really been super successful compared to what we'd hope. And there's also, no one really knows whether these plaques and tangles are causing the disease, or whether they're just a byproduct of the brain trying to, tidy up bad things that are going on. So, in my opinion, I think more basic research is needed to really understand what's driving that pathology. But of course, we need to start looking at treatments now.
It seems to me to be a little bit where they were going with this. They're actually talking about the fact that it's only by getting rid of the plaques and tangles that you could maybe see whether there was a cause and effect relationship between plaques and tangles.
Yeah, that's a good point. Yeah.
So,
Very interesting. Yeah. I don't know. We're not associated with that at all.
Which is interesting.
Yeah, it is. It's very interesting.
You made the comment about brain donations, and it's a question that comes up quite often.
Yeah.
I'm sure you've had it before. The people who are asking about it, what sort of responses do they get, and what brains? You talk about younger brains.
Yeah, well,
Yeah, it's always
we do have
difficult,
brochures.
but yeah.
they gave us brochures to bring along in case we wanted to ask about brain donation.
So we have Marika Asash, who's sort of our technical manager of the Human Brain Bank, and it's sort of a lot through her, but we can leave a few of those.
The
with
process
you, but
is the process has started usually in the will making process. So it's something that you can sort of put into your will that you would like to donate your brain, but at the end of the day, it actually has to be your family that agrees to do it. You know, if the family says no, then there's nothing.
And it's sort of
happens
at that point, like when the time comes, it is the family who sort of have that final decision that they make.
From all over. Yeah, we have donations from all over the country. They can be couriered up to us. We also have donations from the hospital. The shorter the time between the person dying and the brain coming to us, the better, because we can do things like growing those neurons and growing those cells that you see moving at the start. The shorter the time, the better we can do that. So it's quite all hands on deck when it happens. Yeah. So if anyone wants to know more about that, we'll pass on some info.
We'll just get in touch with us and we can, connect you up with Marika to be able to give you more information as well.
Well, thank you very much. I must say it's reassuring on one of your early photos to see that laboratory scientists still wear white coats and have pens in their pockets.
They call it health and safety, but it's just fashion.
We lost ours long ago. That's what you've seen today is just a tiny taster of what you guys do. And the Brain Research New Zealand and the Centre for Brain Research is at the forefront of international research. It's a really well-reputed organisation internationally. There's not many Alzheimer's or meetings that you would go to internationally, and there wasn't something being presented from one of your groups somewhere. So it's some fantastic work. Presumably across the country, you have hundreds of researchers involved in some work. So it's really nice that you guys come out and give us a taste of that, but also and thank you for sort of relating it to the real world of the meetings as well. It's a nice mix. So thank you very much. So on behalf of the people here, so thank you very much for making the time to come.